All the cells involved in immunity are derived from a single type of stem cell via in vivo or in vitro cloning. They are self-renewal cells in the bone marrow that give rise to lymphoid stem cells and hemopoietic stem cells that are both made in the bone stroma. Lymphoidic stem cell will produce three kinds of cells that are T and B lymphocytes and natural killer cells that remain in the blood. And when going towards the tissues, B lymphocytes divide themselves into plasma cells, while the other two remain the same. Hematopoietic stem cell, on the other hand, will produce megakaryocytes. They produce platelets in the blood. Now, ES or erythrocyte cells that will form erythrocytes. GM precursor will lead to the formation of monocytes and granulocytes, namely neutrophils, eosinophils, and basophils. Remember that neutrophils and eosinophils will remain as such in the tissue, while monocytes form dendritic cells and macrophages, while basophils will form mast cells. Now let us start the details from the bone marrow. Unlike most other tissues or organs, the hemopoietic system is constantly renewing itself. In the adult, development of hemopoietic cells occurs predominantly in the bone marrow. In the fetus, before bones develop, hemopoiesis occurs in the yolk sac and then in the liver. Talking about self-renewing stem cells. They are also called totipotent and self-renewing marrow cells. Stem cells are found in low numbers in blood as well as bone marrow, and the numbers can be boosted by the treatment with appropriate growth factors. For example, granulocyte colony-stimulating factor, which greatly facilitates the process of bone marrow transplantation. The stroma contains epithelial and endothelial cells that provide support and secrete growth factors for hemopoiesis. The potential of individual stem cells to give rise to one or more types of hemopoietic cells has been explored by isolating single cells and allowing them to divide many times, and then observing what cell types can be found among the progeny. This process is known as cloning. A clone can be described as a set of daughter cells all arising from the single parent cell. Evidence suggests that in certain conditions, a single stem cell can give rise to all the fully differentiated cells of an adult hemopoietic system.